In Exercise 3, you will begin to perform staining techniques known as differential stains. Differential stains are an important tool in the microbiology laboratory. Differential stains allow an observer to differentiate or separate cells based on staining differences. Differential stains allow this separation by highlighting structural variations between two types of cells or between parts of the same organism. Differential stains permit the observer to see cell size, cell shape, and cell arrangement like simple stains do, but unlike simple stains, they also allow us to gather information about other features. For this reason, differential stains are more commonly used in the lab than simple stains are. Differential stains make use of more than one stain. The first stain used in this staining procedure is referred to as the primary stain, while the second stain used in the procedure is called the secondary stain. It is important as you proceed through the various protocols that you use the stains in the proper sequence indicated in the protocol to get accurate results. Some examples of differential staining techniques are the Gram stain, acid-fast stain, Capsule stain, Endospore stain, and flagellar stain. Let's briefly overview each of the differential stains you will be performing in the lab. The Gram stain is the most commonly used differential stain. The Gram stain separates bacterial cells into two groups based on their cell wall structure. Gram-positive bacteria stain purple during the Gram stain and have thick peptidoglycan cell walls. Peptidoglycan is a special carbohydrate found only in bacterial cell walls. Gram-negative bacteria stain pink and have a thin peptidoglycan cell wall with an outer lipid membrane. The acid-fast stain is a differential stain that separates cells into two groups based on the presence or absence of mycolic acid in their cell walls. Acid-fast cells contain mycolic acid, while non-acid-fast lack mycolic acid. Mycolic acid is a waxy lipid that we will discuss in more detail when we get to the acid-fast stain. The capsule stain is a differential stain performed to determine if a cell has a capsule present. A capsule is a structure located outside the cell walls of some bacteria. The capsule is composed of polysaccharides or a combination of polysaccharides and protein. Capsules help prevent the cell from drying out, and for pathogens, helps prevent phagocytosis by the immune cells of the host. The endospore stain is performed to detect the presence of endospores. Endospores are special structures produced by certain genera of bacteria, such as the genus Bacillus and the genus Clostridia. Endospores are dormant structures produced by cells during hostile or unfavorable conditions. The formation of an endospore is a defensive strategy that serves to protect the DNA of the species. Endospore formation is not a form of reproduction. Instead, one cell will produce one spore. The staining procedure allows us to differentiate between the vegetative cell that produces the endospore and the endospore itself.